After death, no heartbeat, no brain activity, no breath. That seems pretty final. Dead genes. But at the cellular level, only some cells are truly dead. Some cells are still functioning. Scientists want to understand what happens in these lingering cells. To do this, researchers examined the activity of over 1,000 genes in zebrafish and mice for up to 4 days after death. Gene groups related to immune response, inflammation, stress response, and cancer became more active. Even more surprising, genes typically only active during development also suddenly became active. These genes are tightly shut down by cells after embryonic development and are never expressed again, until perhaps the organism dies. So why do they reactivate? Why do they come back to life? It might be that some conditions inside the cell after death resemble those before birth, but this is not certain. Another reason for the activation of developmental genes could be changes in epigenetic regulatory genes. These genes control gene activity by restricting access to DNA. Some of these regulators have been shown to increase activity after death. Developmental genes are genetically locked away, but if regulators spin out of control, these genes might suddenly become accessible and start expressing. There is no real why question here. It doesn't seem like there's a purpose for these genes turning on. That's because genes in a dead organism cannot be acted upon by natural selection. There seems to be no evolutionary advantage to turning them on after death. If an organism isn't completely dead, ramping up some genes, such as those related to stress responses and injury recovery, might be advantageous. However, there isn't enough data to know for sure, and few studies have tackled this question. One study looked at human tissues. Over 9,000 samples from 36 different types were analyzed. After death, gene expression in brain and spleen tissues remained relatively stable, but genes in muscle tissue went into overdrive. More than 600 genes either increased or decreased their activity, and these increases in gene expression fluctuated in a way that suggested biochemical machinery was still working. What can we learn from this? Patterns of gene expression could be used to estimate time of death, something that current forensic techniques don't always get right. Additionally, the study examining human samples identified the types of tissues that might be most useful for forensics: skin and the layer of fat directly beneath, and that's not all. As we learn more about gene expression after death, patients receiving organ transplants from deceased donors might receive better care. Changes in gene activity could even help explain why some transplant recipients are at an increased risk of cancer. It's also important for researchers to understand gene activity in tissues that can only be obtained after death. If death alters our genes, scientists need to know how these changes affect the tissues they study. And simple curiosity also drives this research. Understanding what our genes do after death is a fascinating mystery, and one we have only just begun to explore.